Okay. Good afternoon, everyone. May I have your attention? Our speaker today is Dr. Michel Meunier, who obtained his engineering and master's degrees from the Ecole Polytechnique de Montreal. He also received his PhD in material science from MIT, and is now professor of engineering at the Ecole Polytechnique de Montreal, and also holds a cross-appointment with the biomedical engineering program there. He's been involved in laser processing of various materials and nanomaterials. He has been awarded an National Science and Engineering Research Council of Canada Synergy Prize in 2006 for his fruitful university-industry collaboration with L-Trim Technologies, a company he co-founded. He is a fellow of the Canadian Academy of Engineering, fellow of SPIE, and also of the Optical Society of America. Please welcome our speaker.
Okay, do you hear me? Yeah? Okay, thank you. I've been told that I have to put this on. Usually I don't have a microphone because I speak loud. Okay, so thank you very much for the presentation, and thank you for the invitation to give a talk here at the seminar at Dartmouth University. The subject of the talk is Plasmonic enhanced femtosecond laser cell nanosurgery. Maybe there are some words that you don't recognize here. I mean, probably the word Plasmonic, which is maybe one of the key topic that is being developed now in nanotechnology, in nanophotonics actually. And femtosecond laser, I'll be talking a little bit about how to use femtosecond laser to do a surgery on cells. And the idea here is to be able to do a surgery in a cell, in a living cell, and at the end, the cell is still living. And to be able to do that on a nanometer scale. And what I'm going to present, essentially, is a new tool, is a new scalpel, is a new nanoscalpel to do surgery on cells. But actually, what I'm going to present is much more than a new nanoscalpel. Is a multiple nanoscalpel that can do surgery at many places at the same time on a nanoscale. So this is the idea that I'm going to present. Just to say a few words about where I come from. I mean, this is a 3-hour drive from here. Polytechnique in Montreal, in Canada. It's probably one of the largest engineering school in Canada. I mean, there's a Toronto and Montreal, and so one of the largest. And we may say that this is probably the largest French-speaking engineering school, engineering faculty in North America, probably. So we can expand this and get to do very large distances. So 6,000 students, 4,000 of the bachelor degree, 2,000 master and PhD, 900 graduates per year, 220 professors, and I mean, long time of history, 135 years. A few words about what I'm doing as a researcher. I'm doing research in the field of laser processing and plasmonic laboratory, where we develop and model new laser processes of materials for nanotechnology and biomedical, and we develop new plasmonics nanostructure for biophotonic applications. This is a picture taken last year at this time. So at any time, there's roughly 20 people in my research team, roughly 12 to 15 graduate students, and some research personnel and some undergraduate students as well. Oh, the other way. Okay. Research activities in my group, the key words are femtosecond laser and plasmonics and biomedical. So I'm coupling optics with nanotechnology and application in biomedical. This is basically what I'm doing. So a few words about the various subject that I'm covering here, femtosecond laser or femtosecond engineering, where we use femtosecond laser to micromachine materials, and we do that from the simulation point of view as well as from the experimental point of view. So my background is, as has been told, my background is in material science, actually. So using laser to process materials. And this is basically what I've been doing in the last 30 years. So this is one topic that I'm doing. The other one is that because we are able to fabricate materials that are very special, we started to use these very special nanomaterials to do application in biomedical application. So the fabrication and characterization of plasmonics and magnetic nanoparticles. We are finishing a project now. We can fabricate nanodrugs. We can fabricate drugs that are on a nanoscale with a laser. This is not the topic I'm going to cover, but I can answer a question about that later on. Plasmonic is a term that I'm going to maybe to explain a little bit more later, but we can use plasmon, which are collective excitation of electrons in a metal. We can use plasmon to do biosensors, and biologists are using that for many years. It's called surface plasmon resonance. It's a SPR. So biologists are using that for many, many years, and what we are developing in our lab, there are new ways of doing plasmonics with nanotechnology approach, SPR, and to do SIRS, which is surface and Raman scattering, as well as to do that to detect very, very little and highly sensitive concentration, very little concentration of virus, and also for cancer detection. The subject I'm going to talk about today is the one on laser nanosurgery, using femtosecond laser, using plasmonics. We do fundamental and simulation and application to cancer therapy. This is something that I'm going to cover because I thought it would be a good overlap or some overlap with some of the subject that has been covered here in biomedical application. So the content. The idea would be to develop a new tool. And what I'm going to cover is a tool of the future, a tool that can do a surgery on a nanoscale using a laser and using nanotechnology. So nanosurgery with a nanoscalpel. The idea would be to be able, with a scalpel here, to perform on cells. This is these are cells, by the way. I'm not sure if this is the good representation anyway. So cells with a nucleus here. So we can do very fine surgery, and this surgery you can do many things with that, many applications, and I'm going to cover that in this presentation, is to do transfection of cells. This is what I'm going to cover as an application. But essentially, this technique is probably much more diverse in terms of application. We can use that as a new way of performing a surgery on a nanoscale. So this is this nanoscalpel. And actually, what I would like to do, much more than that, so why not that we can use the same approach and do, let's say, a surgery on these cells, but not the other ones, just these cells. So if I can target something, which are nanoparticles actually, that will go to a specific cell and not the other one, and use a laser, I can essentially do multiple nanosurgery at the same time. And this is essentially the approach that we would like to develop. So this is a new tool which can do nano at the same time that multiple way of doing surgery. Okay, so these are the content. The content I'm going to cover here is to tell you about femtosecond laser plasmonic enhanced nanoprocessing. I will tell you why the plasmon arrive in this there. We saw that on materials. We started with an observation on a material actually, and we switched that to the application to biomedical. So essentially, this is the observation that we made. This is the idea of the multi-nanoscalpel. We apply that to transfection of cells. The important thing is that I'm going to cover that is the nanoparticles that are there are not broken. This is very important. This is an efficient technique to do cell transfection. And the last two topics are essentially more fundamental. I'm a physicist. I'm interested in how it works. And so the interaction of a laser with the nanoparticles in a biological media is to understand how this interaction occur, and how can we make a nanosurgery. And this is the basis of that from the experimental point of view, and the last topic is from the more fundamental and theoretical point of view. I'm not going to go into much detail on that. These are the last few slides, but actually it gives us the idea about how it is working. Okay, the other way around, I will learn it. Okay, introduction to femtosecond lasers nanosurgery. I don't know if there are some people that are old enough than me, old enough like me, not than me, sorry my English, to remember the film called The Fantastic Voyage. Who remember Fantastic Voyage? Usually, I mean, the young people, they don't remember that. Fantastic Voyage, that's a film in 1966. Invention of the laser, they use laser. Invention of the laser is 1960, 61. Okay, so that's been 50 years of the invention of laser. And actually, in this film here, what they do, I took it. Do you hear that? Yes.
Listening to the sound of a completely new screen experience.
The exact word is not so important, but just to tell you here that in this film, for those who have not seen it, to remove a blood clot in the brain, they miniaturize a submarine, and then they put the people in the submarine, and they say, okay, we're going to go into this brain in this submarine here, arrive at this place, and then use a laser. And you use a laser to do a surgery, a local surgery with your laser. And your laser was very new at that time. And when I was a teenager, I was very impressed by this film. I don't know if you were, but it seems that apparently they're going to do a remake of that. This is what I remember. And I don't know for those who remember, I mean, Raquel Welch. I don't know, remember Raquel Welch? You remember Raquel Welch? That's some sort of sex symbol at that time. Okay, and in this film, it's very strange because she's tuning a laser, and it's very good. I mean, pulling around the lenses and so on. Usually, when I have more time, sometimes I show this film, but I don't have too much time. But essentially, that's very nice. Anyway, so the idea here is to use laser. To use a laser that was, of course, introduced by remains, I mean, more than 60 years ago. This is the laser we're going to use. It's a femtosecond laser to do this surgery. It's surgery on a very fine scale. And the idea of using femtosecond laser is, from my background in material science, in material engineering, this is the type of laser that if one wants to do a very fine machining on the material, you use a femtosecond laser instead of nanosecond laser, because of the fact that you have a limited heat-affected zone. You have a limited diffusion of energy for that. The idea, this is a very well-known picture done more than 15 years ago. This is a piece of metal, essentially the same conditions. Femtosecond laser, you do a very fine machining, and nanosecond laser, you have a lot of material that's been melted, and because the interaction time is very long. And if it is very long, there's some energy that has time to diffuse away and start melting and so on. This is the basic idea about that. So the idea now is to use this laser to do a fine nanosurgery in living cells. And also the advantage of this laser is the laser wavelength is 800 nanometer, and this is in the weakly absorbing window of the biological media. So this is pretty interesting. So actually, this can go deeply into biological medium, and resolution can be on the order of the wavelength, or something like a micron, or maybe a little bit, I mean, on that order of magnitude. It can be better if we consider nonlinear effects and due to multiphoton absorption. But actually, the effect is sometimes much larger in terms of that. My colleague, Eric Mazur at Harvard, did that some years ago on living cells and do some kind of surgery at different places and show that here you can cut different places. Like here, you see this region here is around few microns. This is 10 microns here. This is few microns. And he is using that to understand the basic mechanism in cells. You have to collaborate with biologists that will tell you, okay, I'm going to look at that and that and that. There are some basic phenomena occurring into the cell. So you can do neurobiology and so on. So there is a lot of research using this tool, this very specific tool. So this is much more like a microscalpel in a sense that I'm shining the laser and I'm making some kind of hole, a specific hole at a different place, which can be roughly, micron, a little bit larger than micron, maybe 2 microns, depending on the energy that is being used. And for that, it is essentially like a low throughput. I have to focus on a specific place and do it, and then another place and do it, and so on and so on and so on. Which in some application, the good way to do. But in some other application, you need much higher throughput. If I can invent something that can do this on a nanoscale, but at different places at the same time, that would be very interesting. So in parallel with that, we saw an interesting observation. We were not alone of having seen that. But the idea was coming from a very different field. It's in the idea of nanoprocessing. It's processing on a material. So the idea here that what we saw is that we put like nanoparticles on the surface and shine a laser. So experimentally, you have silicon. I'm not sure if this is the silicon and this is an SiO2. You can clean up the SiO2. I'm not sure if you see that from the back of the room. Here, you don't need, Alex, you don't need it. This is not so critical, I think. I don't want to speak in the dark. Because the people will start sneezing. So if I put some gold nanorods, in this case it's a nanorod, it can be a nanoparticle, and these nanorods can be really controlled. This is gold nanorods, and in the field of nanotechnology, these nanostructure that we call, you can control that and very finely control the dimension of these rods. This is 87.5 nanometer per 41 nanometer, and this is a silicon substrate. And then you shine. These are the nanorods on the surface, and then you shine a laser on it, and what happened is that there is a little, I'm not sure if you see it from the back, there is little places here, there's little ablation, very fine holes, and actually, this is a picture after that, but if you do an AFM, this is an atomic force microscope, to look very finely on this hole, one of this hole, what you will see is something that is very, very small here with a certain region around it, but there is a region where it looks like this is a hole that looks like the imprint of this nanorod in this case. So this is pretty interesting. So what we can do here is that we can with a nanoparticle, with a plasmonic nanoparticle, essentially amplify the field so that I can do a highly localized process, a highly localized ablation using this nanoparticle. This is due to Plasmon. So a 1-minute course lecture on what is a plasmon, because this is a very important point, very important topic in this seminar. A plasmon is a collective free electron oscillation in a metal, and as you use these nanoparticles let's say in a liquid, if you put gold as a nanoparticle in a liquid, it will look red. It will look like this here. So this is that depends on dimension, actually. So this is gold nanoparticles. Gold looks like gold, as we know, when it is bulk, but when it is on the form of a nanoparticle, it looks like red like this. It is red because of plasmon. Because there is this plasmon peak arriving here at around 520 nanometer, and then there is nothing here. And if this particle is in a liquid, when you shine the light through it, the red color will go through it, and the absorption of all the green and so on will be absorbed. So it would look red. So this is the, my 1 minute course. Okay, I have another slide on that. So the important phenomena of a plasmonic structure like this is that you have a field amplification around it. And this is a nanoparticle, and this is the calculation if you just use Maxwell's equation and you solve it. What you're going to have here is a field amplification. So if you irradiate with 800 nanometer with a polarization like this, you get a field amplification that can be as high as 4.5. But actually, if it is a nanorod, it can be actually in resonance at 800 nanometer, and the field amplification can be much higher, and 75. So the idea here is that there is a field amplification. So if you put a nanoparticle, something on the surface, and you shine the laser, you have a field amplification close to the particle. And this will give the source of a nano process, but just close to the particles, and it dies off very rapidly. As you see here, this is 100 nanometer, and away, few nanometers away from that, I mean, the field is very small. It is decreasing. So the surgery can be on a very nano scale. Okay, so if one uses this, like I turn it because I want to show you that, this is the field amplification of a nanorod. This is the imprint, let's say, of a nanorod over the ablation. So the idea is that by shining with a femtosecond laser on a nanoparticle like this, this will amplify the field around the particle such that I can produce a nanoablation. And that's the idea of doing this nanosurgery on a nanoscale. So the idea now is this multiscalpel here. So the proposed approach is that. So remember of the presentation of the fantastic voyage, the small submarine going into the body and arriving to a place, I'm going to replace that by a nanoparticle that is going to a specific place, and then the laser comes from outside in our case, but and then there will be an amplification close to the particle such that I'm going to produce a nanoscale ablation. So I'm going to use here, as a general approach, nanotechnology, where you have here nanoparticles with some, maybe some magnetic nanomaterials, some optical materials, and also some chemistry targeting on the surface, such that this particle will go into the body and arrive to a specific cells. If it bounds at a specific place, that's where the surgery will happen. And that's the idea of using that. So the idea is the following. We use plasmonic nanostructure that will locally amplify the field, producing a highly localized process on a nanoscale. And then the dislocation is decided by biofunctionalizing the nanostructure. I need a good chemist with us that will put the chemistry on it so that it will arrive to the specific cells. And then, by shining the laser, now I don't focus the laser. I just send the laser large, and the nanoparticle will focus the energy. And at the place where there are no nanoparticles, there's nothing happening, because at 800 nanometer it's transparent. But at the place where the nanoparticles are, there will be a specific surgery that happens. So plasmonic nanostructure that will bind due to some functionalization here, and then after the irradiation, there will be a specific ablation or whatever. Okay, so the idea of the nanoscalpel is now to take this nanoparticle, arrive to a specific cell, from the chemistry point of view to arrive to a specific cell, and then from that, shine the laser. Now the laser is not focused, it's very large, but the energy is concentrated only at the place where I put the nanoparticle. So I can produce a very fine, on the nanoscale, this the very fine interaction. And I'm going to show you that I can produce that on the hundreds of nanometer, that I can do it in the living cell. Actually, if I target it to a specific cell, let's say cancer cell, I want to target some cancer cell, but not the other, the cells that are good. So if I do that with the particle, and then the energy will arrive, there will be like some sort of a multiple nanosurgery at the same time. So a multiple nanoscalpel that will do the job. Okay? So what it is good for? I think that this new tool is a tool that can be used for many types of application. I'm going to show you one that we're working for a couple of years now, is to be able to do transfection of cells. Transfection of cells is an approach that biologists are using for many years. Actually, doing transfection of living cells, actually, the idea is to include an external DNA into an exchange the DNA of a cell and to do this gene therapy. So it's gene therapy. And gene therapy has been developed by biologists for many years using endocytosis, using like liposome. My colleague, I'm collaborating with a colleague of mine at Meunier University. This is what he's doing. Actually, you can do some injection. And one of the most popular one is to use a virus. So you put the gene that you want to transfect the cell in the virus, and the virus does the job and arrive to a cell and inject the DNA. The problem is, while all biologists accept that the fact that these viruses are not very harmful, it's very difficult to get FDA approved when you do it on a human body. One of my colleagues is doing HIV. They use HIV for doing this. And he is telling me HIV is not harmful at all because I excluded all the DNA and everything, and I'm putting the DNA that I want. But even that, it's not possible to get FDA approved if it's using HIV, of course. This is a particular problem. If you do it in vivo, in human. So this is very difficult, and people have been developing physical ways of doing this. A physical way is using, for instance, an electrical shock. You do you put the cell and you put a high voltage on it so that, these cells are open and they are afraid, whatever, and then the DNA includes into the cell. And the other way is to use light. And this is what I'm going to show you. So the application of gene transfer is very large, using stem cell therapy, tissue engineering, cancer treatment, and this is one of the applications that we are aiming right now is to use this technique for cancer treatment. So the idea is that you have a cell, you have a nucleus here, you have a foreign DNA. The idea is to be able, to open the cell very finely. You don't want. It's important not to kill the cell. You want just to open the membrane, and then you do the transfection on that. So that's the general idea, and this is one application of this nanoscalpel. Okay, so we made a little video here of what we are. Okay, so this is representation of the cell, a surface of cells, and these are particles. So you have gold particles here. This is around the cells, and then it arrives on the surface, on the cells. You have also some exogenous plasmid. So you have some plasmid. This is the DNA that will enter into the cell. So the idea is that this is a representation of what is happening. So you have multiple nanoparticles here, femtosecond laser producing a nanobubble around it. This nanobubble will perforate the membrane, and then these bubbles will last during a certain time. And then, as you, let's say here, I'm incorporating like a green fluorescent protein into the cell would become green. This is a proof of principle of the technique. So this is basically what is presented. So that's the idea that what we have developed here. So we have cells on a in vitro cells. So we scan it here. We scan this part here. We don't scan this part. We just scan this part. And then after different steps, and then you can count the number of cell that has been autoperforated, and we use a green fluorophore inside to get the cell green, so that we say that there is a successful optoperation. It's like a simulation of a transfection in this case. So this is high throughput. The important point is these nanoparticles that I'm using do not break. And this is very important. If you want to use a scalpel, you don't want to break your scalpel as you're doing surgery. Of course, it will give you some problems. So what I want here is that I'm putting nanoparticles there. I don't want to break these particles. They will stay intact. And then in the body, they might be eliminated and go into the spleen or the liver or so on. So there's been some study on that, on these particles that will be eliminated somehow. So the idea is that we have to prove that these particles are not broken. So what particles should we use? Should we use particles like this sphere here, where the amplification is not so large, or in resonance, what I call in resonance, where the amplification is very large? So there is a lot of the energy is highly absorbed in this case, and not so highly absorbed in this case. Which one is better? That was the question at the beginning that we were asking, and this is a very important point. If one looks at the absorption here, at 800 nanometer, this is the Plasmonic peak at around 520. At 800 nanometer, it is shifted to 560 or something like that. The black curve is the absorption. The black curve is almost zero at 800, but this is a scattering that is important. This particle would scatter light largely. And this is why we use that in imaging, for instance. We use that because the particles scatter the light, and in the dark field, you will see the particles. Using a nanorod, you can set up the nanorod so that, and we have this type of nanorods that will absorb at 800 nanometer. With a high peak here, actually, if one calculates the electric field amplification, this is around 75, and the one is around 4.5. So in the case of nanorods, the absorption of energy is very large. In the case of nanoparticle, this case, the absorption of energy is very small. Which one is the better? Which one is the better for our approach? It happens to be that the one that is better is this one. I don't want to have too much absorption of the energy, because if I have a lot of absorption, I would break the particle. If I break the particle, the particle will be broken in small pieces, and then I will have problems. It's just that I'm breaking my scalpel. So I don't want to break the scalpel. So that's the idea, that what we show, and we've done that by 12 hours of irradiation of the particles, and if you look at the spectrum, and if there's a change in the spectrum, it means there's a change in the size, and it means that the particles or the nanorod has been broken. And actually, I'm not going into too much detail of that, just to tell you that there is a change when you start having fluence over 0.08 millijoules per square centimeter. But in the case of the off-resonance particle here, it's higher than 500 millijoules per square centimeter. So many orders of magnitude difference. And actually, we are operating at around that value, 100 millijoules per square centimeter. So the nanoparticles are not broken, and this is very important. If one uses this type, it will break, and this is giving us problems. So the good material to use is a nanoparticle. It's not the nanorod. So these are on typical cells. I can put roughly, this is 200 nanometer nanoparticles just to see them. We were able to count them before radiation and after radiation, and essentially the number of particles are essentially the same. It's, so we don't, if it was broken, you'll see many pieces, and we don't see that. So the nanoparticles are not broken, and the nanorods are actually broken, and they are very difficult to observe, actually. Are these particles toxic? We have done some standard MTT tests. The biologists are doing that quite a lot. And basically, with that, what we show, and I don't want to go into much detail about this result, just to show you here that the toxicity of these particles after 36 hours, it's around 1% of these particles, and nanorod is around 9%. And usually nanorods present much more problem in terms of toxicity because of the presence of the residue of CTAB. CTAB is a chemical used for the fabrication of nanorod, and actually it is a toxic chemical. So this is the problem with that. Okay, so we were able to show with that. Let's move here. We were able to show that with that we can do the transfection, and this is here. I'm plotting the viability as a function of fluence, the viability, and here the perforation rate, and actually the perforation rate gives us a value. This is all the value here. So the viability is around 90%. So in this process, even if I'm using the laser shining directly on the nanoparticles on the cell, the cells are still living. So 90% is highly it's a very high value in terms of viability. And the perforation rate is around 65%, which is pretty good, actually. And if we do that with another type of laser, we can cover, here it was to show that we can cover 40 millimeters square in a minute. So covering a large area would take a few minutes, which is not so bad if I want to cover a certain region, which is centimeter square. This is pretty good, actually. And the other point here is the viability of the cells after 24 hours, 48 hours, 72 hours. It is increasing here. And this is the good news. The cells are growing. So in this case, so it means that they are viable and the cells are growing. So the cells that you can transfect them and they can continue to grow. So they are not basically affected over that time frame. And finally, comparison with liposuction. So liposuction is a technique that my colleague at McGill University, Jean-Jacques Lebrun, is using to do transfection of these cells. And actually, liposuction is doing a transfection of around 10%, because this is much bigger than the GFP, the green fluorescence protein. This is much bigger, so it's a little bit more difficult to get it in, so the efficiency is smaller, but it is still 3 times better. So it is a very powerful tool that we are using. So the application is very, very good. So in my last slides, I'm going to go in a little bit more detail how it works. How it works from a physics point of view here. I'm interested to understand what is the phenomena where I'm shining the femtosecond laser on a nanomaterial, and what does it do? At the end, there is some kind of hole created. What are the steps to arrive to these holes? Actually, a hole is probably a bubble. It's a small bubble. It's a nano bubble. And this is what I'd like to measure. How I'm going to measure that is by some sort of in situ measurement, optical measurement. And then we'll have also a theoretical simulation to show that we are forming nanobubbles. So 2 of my students, a PhD student, is doing the experiment, is doing the theory, And so they are working together. Okay, the idea to measure the nanobubble is the following. So we're not considering cells now. We're working on nanoparticles in water. And biological media is very close to water, actually, in terms of this process here. I mean, for an index of refraction and so on, it is very similar. That's why we have done most of these experiments in water. So let's say I'm shining a laser. This is the probe laser. I'm shining the probe laser through the content here. It goes to a lens and goes to a pinhole and a detector. So we detect a signal. And if something happened here, if there is something happening here that is changing the index very locally, what will happen is that the probe will go and will have a different optical path that will go outside this pinhole. And of course, what will happen here at the detector will have a lower value. So what I'm going to measure is something that is going down, and then when this disappears, it will come up. So this going down and going up is something that I'm going to measure. And this is related to the presence of something happening here, something happening in the liquid. So this is actually the way to measure some sort of a pressure wave as well as the nanobubbles. So the system is the following here. We have a femtosecond laser here that will go directly here. And we have an helium neon laser, a very conventional CW laser that is going through here and then going through the pinhole. This is the pinhole. This is the detector. And the signal will arrive here. So basically, after I'm creating this nanobubble here, after the nanobubble, there would be a signal going down. And if I can measure this signal correctly, I would be able to deduce what is happening here. So this is the result, typical result, where we see here at the beginning, this is on a microsecond scale. This is a shock wave at the beginning, because at the beginning what will happen is I'm shining the laser close to the particle. There will be a shock wave at the beginning, and then a bubble that will arrive. So these two phenomena are closely related, actually. So one is the first one. There will be a temperature rise, and then a shock wave, and then a cavitation bubble. This is a cavitation bubble because when you create a bubble, it will be a bubble created. At some point, it's too large, and then it will collapse. And then it will start again, and then collapse. So what you see is an oscillation. So you see this oscillation here, oscillation. And so this is the signature of a cavitation bubble. So if you go in much detail here, what you see here, and the important point is that this is only water. Water here in orange. What happens is that when there's only water, there's almost nothing happening. But when I put nanoparticles and nanorods, there will be a lot of phenomena happening here. So the point is that these nanoparticles will enhance the generation of shockwave and bubble formation. So the nanoparticle is the source of this fabrication of the nanobubble and the pressure wave. So the idea is it pressure wave, actually? So if it is a pressure wave, the speed at which the speed at which the pressure wave will evaluate into the liquid should be the speed of sound. So the way to measure that is to do the following. This is the probe beam. If I do it here, there will be like a pressure wave and a signature here. If I move this to a certain distance, d, and I do the same thing, it will happen, but in a time scale, a little bit off here. So the difference in time here, this is a time scale. This is the time, and this is distance. So you have distance divided by the time to give you the velocity. So if one does it correctly, and I don't want to go into much detail about all these results and so on, but you plot this here, the distance versus time, the the slope here is the speed, and this is the speed of sound known in the literature, and this is the speed of sound that we measure. So actually, this is a real speed, real expansion of at the speed of sound. So this is a pressure wave. Okay, so the other thing is to measure the bubble. So the bubble, there is this oscillation, but actually this oscillation is with higher fluence. At lower fluence, we see only one of this. And there is no oscillation. There's only one. And from that, if one looks at the bubble lifetime, these are the numbers. And if one look at this number here and through a model, this is not direct. It has to be done with calculation. But we have done this, and we were able to calculate the bubble diameter, which is, in this case, at 200 millijoules per square centimeter is around 800 nanometer. So the bubble is around 800 nanometer. But I can do much better than that. If I go lower in terms of fluence, at 200 millijoules per square centimeter is a little bit higher, and then it goes down, we can arrive to something like 200 or 300 nanometers. So we can do a nano bubble at the place we want, and with a very highly controlled by controlling the fluence. Okay. So we know that we're producing a pressure wave. We know that we're producing a nanobubble. This nanobubble, that's the source of this nanosurgery. So from that, from the basic physics, and this is the more simulation and theoretical point of this problem here, of this process. What we want to understand is what's going on when the laser interacts with a nanoparticle. So many things can happen. So this is the particle. This is the field amplification. What can happen is that this particle can be heated by the laser because of very tiny absorption, but still can be heated. You can also, due to this heating phenomena, heat the water around, and this water being heated will change its phase and start to become a bubble. So this is one way that we can think about the fact that we can produce a nanobubble. But this is not the way that it is in our case. What is actually going on is that due to the field amplification here, there will be here electrons that are excited and producing a nanoplasma. So this is a plasma produced very closely to this particle. Electrons are excited directly in the water. Because of field amplification, you get to the point that you do an optical breakdown into the water. And this optical breakdown will release its energy directly in the water without affecting the particle. So there will be here water that's being heated, and this is so rapid that will produce a pressure wave because you have a very sudden increase in temperature of the water. We're talking about nanoscale here, in the tens of nanometer around the particle. This would produce a pressure wave, and finally, this pressure wave would generate a bubble that is on a nanoscale around the particle. So these are the various steps. Actually, the idea of the off resonance is very important because you have low absorption that avoids the fragmentation of the particles. Absorption is very small. This is the blue line here. Absorption is very small, but actually the near field amplification is sufficiently large that will lead to a plasma generation and cavitation. And that is the way that the process works. And actually, because of this low absorption here, the particles do not break. But it produces enough nanoplasma that will generate this cavitation and nanobubble close to the particle. So we were able to model this using finite element approaches. It took the student that is doing his PhD in physics almost 3 years now. That involves 10 differential equations, all coupled. It's a very complicated problem, because you have a time scale that is very fast here with the electromagnetic irradiation. Plasma production, it's called two-temperature model. I don't want to go too much detail on that. And water, what is going on in water is Navier-Stokes equations. So it's a very complicated and all coupled and strongly nonlinear. We were able to do that. This is the plasma density. So showing that we can produce a plasma that is very close. Polarization is horizontal, so plasma is here. And this plasma gives rise to a pressure, like so sort of pressure wave. It's like a tsunami. I was giving this talk in Japan, and I realized that I shouldn't use the word tsunami. But this tsunami that is produced is highly localized and horizontal, actually. So it goes in the same direction in the polarization. And then, after that, we arrive with the production of a bubble. The bubble at 200 millijoules per square centimeter, this is the value that we did experimentally. Actually, we arrive with a value that is very close to the experiments here. This is simulation and experiments. The values are very close. We're very happy about that because, I mean, calculation, it takes, I don't know, 1 or 2 months of calculation on mainframe to arrive to that. And of course, the experimental result takes, microseconds to get. But they are very good in terms of agreement between the two. What we can do with model, now we have a model. What we can do with the model is we have buttons that we can turn off and on. And what we have done is to turn off the plasma. If we turn the plasma off, we can see what happens. And this is what we did here. So in the calculation, we just turn plasma production, and we do the calculation. And what happened is that there is no bubble. So if there is only, and of course here, there is heating here. Heating is there, but plasma is not there. So there are no bubble. So the conclusion from that is that no bubble are formed without the plasma. So the plasma is the main source of the production of particle. And this is a new way of producing a nano bubble into the biological media. So the numbers here are very close, 800 nanometer at 80 nanosecond, and this is the simulation, this is the experimental results. Very good agreement between the two. We're very happy about that. That's very, very good. Okay, so what is going on in terms of on the time scale? So the laser interaction, this is on the time scale here. 10 femtosecond, 100 femtosecond, 1 pico, 10, 100, etc. The pulse width is in the range of tens of femtosecond. Electron heating into the material, into the goal, is on that range. There is also some thermal emission because of the heating. I don't want to go into much detail about that. I can answer a question. But, and there is a lattice heating into the particle that starts in the hundreds of picosecond. And then there's a plasma dynamics. Plasma dynamics starts because of this field that is amplified close to it. So there is an impact ionization, photoionization, and then plasma expansion that starts in the hundreds of picosecond and expanding. And then the energy is transferred to the water, producing through water heating and the pressure wave, and then finally the cavitation that starts in the tens of picosecond. So this nano bubble starts in the tens of picosecond, and tens of picosecond, and will last in the nanosecond range. For transfection, if you have a membrane and you produce a nano bubble in the membrane, it will like perforate the membrane during nanoseconds, but then the bubble disappears, but the membrane stay open during a much longer time, actually. My colleague of mine in Germany, they are doing some experiments on that, and it arrives to many microseconds for the first closing, and then it can even last even many seconds or minutes because of the obturation of the cell. So this process is much longer, but for the biological point of view, it's longer. From the physics point of view, everything is finished in the nanosecond range. So so this is the representation here. The pulse gives you some sort of a amplification of the femtosecond laser near field by close to the particle, and then the production of a plasma. The polarization is horizontal here. 100 femtosecond generation, a heating of a plasma that will expand. It will expand up to hundreds of picoseconds. And then the pressure wave will start because of this plasma that will release the energy and gives the energy to the water. We start producing at the beginning a pressure wave, a fast relaxation of the plasma that hits the water and releases the strong pressure wave. And then finally, the cavitation that will start in the tens of picoseconds up to many nanoseconds. So that has been submitted to Nature. Photonics, we're still arguing with the editor. Anyway, this is Nature is not, they are very difficult, but I think we're going to win on that. So conclusion, we introduce a new way to do nanosurgery. It's a surgery, it's a new tool. It's a new tool for the 21st century, I think. It's a new scalpel on a nanoscale, on living cells. I can crank up the laser and kill cells if I want, or I can do this nanosurgery very finely and still get the cells living. I show one application of this multi-nanoscalpel. Optoporation and transfection with 70% of optoporation and the cell viability over 90%, and the efficiency with some of the competitor in terms of the way of doing transfection, 3 times better on these type of cells. From the physics point of view, from the mechanism point of view, we show that these nanoparticles are not broken. This is very important. I don't want to break the scalpel. So I'm not breaking the particles. The particles are still there. We can detect the shock wave and bubble in that range here, in this case. We can have bubbles smaller than that. We can go in the 200 nanometer scale. There is it's because it is sometimes difficult to measure. But in the range that we're doing optoporation, this is the orders of magnitude. So the model is there is a near field of resonance amplification that gives rise to a plasma. The plasma gives rise to a pressure wave and cavitation, and that gives rise to the cell optoporation. We detail the physics here, and if I cut the plasma, I'm cutting this. So the plasma is very important. The nanoplasma is important. It is not a heating phenomena. It's a plasma production that gives rise to the pressure wave and nanocavitation. What is from here to there, we're still working on it. There's a lot of unknown here on this application. We're not really sure about all the details. That involves a lot of biology, actually. I'm not a biologist, I'm a physicist, so I'm detailing this part here. So that was the way of doing surgery in the past, and probably this will continue for quite a long time. And this is a possible way of using nanosurgery for the future, if one wants to use that in other applications. We've shown that we can use that as an efficient transfection technique. There are certainly other applications. I am working right now on another application, or 2 other applications, and we don't have results yet, so I'm not going to tell you the detail, but I will be very, very pleased to come back and tell you that when they have more results on that. But I think that I have a very nice application of this. But I'm looking for other applications. I don't know if someone has some ideas about how to use that. I mean, of course, this is nano. It's not micro. And most of the time when you do surgery, it's micro and probably even millimeter range. That's what I understood from Frédéric. So surgery is certainly much larger. But in any case, I think that performing let's say, in some sort of living cells, if I can take these nanoparticles and arrive to a specific region, like a DNA strand, and then use a laser, I can cut at a different place. I can cut at many at the same place at the same time, many places at the same time. That would be very nice. I'm not sure about the application. I mean, people in biology and medicine should tell me if there is other application of this new tool. I'd like to thank the work of, this is a photo of my group last year, and Judith Baumgart did a fantastic postdoc with me doing this, all the transfection. She was very good, really, and so she is now back to Germany. Rémi Lachaine is the person doing all the physical and the in situ measurement is very good to do very complicated experiments. He is with the femtosecond laser. Alexandre Robitaille did a lot of work on on materials. He is not directly doing on biology, but he clearly showed the formation of nano ablation and also the physics. I didn't have time to talk about that, but this is a master's student, and we just submitted a paper that he just did to Physical Review later. This is a very, very nice work, actually. So, and Etienne is doing all the theory, and this is the key guy that understands everything what is going on. And I think that his model is pretty fantastic. He successfully put all that together. The rest of my team are doing other stuff related to this, the Plasmonic and laser and stuff like that. I didn't have time to go all that, but I certainly, I prefer to talk only one topic instead of talking about many topics. So I'm going to stop here. I think that I was a little bit late. I'm at 2-3 minutes late. I think I'm okay. I thank you a lot for your attention, and I'm welcome questions or comments. Thank you. Yeah, there was a question in the back.
Yeah, so what would be the limitations to actually performing a surgery, say, in a human or even
To what?
To performing actual surgery in a human, like how deeply can the laser penetrate?
At 800 nanometer, I mean, there are, centimeters or something like that. Maybe Frédéric has a much better number, or 2 centimeter. Yeah, it's so basically this is feasible. I did not really mention in this talk. I forgot to mention the cells that we're looking at are melanoma cells. It was written, but I did not emphasize that. So they are melanoma cells. So they are on the skin. So the idea of the application we have in mind, and this is actually from my colleague at the McGill University, we want to do transfection of cancer cell to change their DNA content so that they don't migrate easily. So that's the application. Of course, I can crank the laser and kill all the cancer cells, and this is certainly a way to do. So there are limitations in terms of in vivo, but if it is, let's say, open, or if I was discussing with Frédéric about neurosurgery, using nanoparticles at different places and to do surgery with laser. I don't know if it is, but this would penetrate, a centimeter. So that in that case, yeah, each application would have to do some limitation. Most of the time we do it in vitro. And in vitro, there are only one layer. So this is not critical. That answer your question?
Yeah, absolutely.
Okay.
I had a question regarding the efficiency. The nanoparticles are not spherical, or completely spherical, are they?
The nanoparticles are spherical.
Oh, they are, okay. If you use nanorods, you have an issue of lining the field with the rod.
Now, as I was showing in the talk, a nanorod will absorb the energy, but at some point it would break. And we used that at the beginning, actually. And we realized that the results were not so good. So we changed with the nanoparticles and tried to understand that. Because intuitively, at the beginning, what you want to do is to have energy absorbed into your nano whatever and to do the process. But actually, with the nanorod, it is, you break the nanorod. So as you are scanning, at some point, and we were just breaking nanorods, and they were probably transforming in nanoparticles. Because if you heat up a nanorod, it would transform in a nanoparticle, because energetically, a nanorod is more higher in energy compared to a particle. See, the nanorods are not stable. Yeah, you have to speak loud, and actually I don't see you because I have this spot here.
How do you make sure that the gold particles do not land on a benign cell and you don't destroy that?
I'm not sure if I understand. Maybe you have to speak louder, or.
How do you make sure that your gold particles are cell-specific, that they do not land on a benign cell and destroy that?
How do I measure the particle? I'm sorry, I have difficulties.
Asking if your
What?
if your nanoparticles are doing normal cells as well as.
Yeah, it's because of, in all the experiments I've shown, there is no functionalization. We're working on that. The principle is if we put the particle with a good chemical on it, this chemical will find the cell that I want to do the measure, to do some therapy. And the other cell, it will not bound to it. That's the basic principle. And this is known in the field of nanotechnology using nanoparticles. We haven't done everything that I've shown. There is no functionalization. So all the cells are the same, and I put nanoparticles everywhere. Right now, this is that. The next step would be to show that I can target one specific particle and not the other one. And this is something that we're working on.
If there are any more questions, I invite you to meet with Michel Meunier. He'll be glad to answer. We're done. Thank you very much.
Okay, thank you.